the Institut Pasteur. I'm Guillaume Dumas, and I'm waiting for Pierre-Marie Guédot, who is in Paris. I moved since last week in South Korea. I'm in Institut Pasteur Korea in Seoul. So I'm gonna start to answer some of the burning questions you asked on the forum. There are many. I will let some from Pierre-Marie if he come, or if he can't, he's gonna answer next week. First, a very amusing question that seems a little bit futuristic, but is a way of designing synthetic olfactory receptors in the near future. It's G.P. Patra who asked this question. So actually, it's already the case. There are people synthesizing human olfactory receptor in order to create biosensing devices. So this is not the future, this is right now. This, of course, preliminary technology. We are far from having a full synthetic nose, but this is the idea to go with. Related to that, we have also a question. I know this one will be for Pierre-Marie. Another question from Christelle Coville about oxytocin and chloride levels in autism. Has anything been proven for other developmental disorders, particularly ADHD? So there are many studies of oxytocin in neuropsychiatry disorder right now. You can go in clinical trials websites and check all those trials ongoing with oxytocin. So it's kind of a hot topic. We already discussed last week that there is a lot of contradictory results though. So a lot of them are still ongoing, and it's hard to give a clear answer about association between oxytocin and a specific phenotype. There have been a lot of studies showing that oxytocin can increase a kind of affiliation or care for others, but also a growing number of studies now showing opposite effects regarding an increase of aggressivity against others, but more like out-group versus in-group. So as usual in cognitive neuroscience, you can observe some opposite effect of the same factors. So it shows nonlinear and unpredictable brain and behavior can be in humans, especially. And in the case of oxytocin, we are here with an hormone which cause many regulatory things, and many have nonlinear effects. So my gut feeling about this is to be very cautious and check for replicability of studies on those topics. Well, we're gonna see in the next years, hopefully, replicability of major results on oxytocin. What do we have? Question by PMUS 112. So this is a very nice question about neurogenesis. So we've seen how the creation of new neuron, especially in hippocampus, for instance, can increase our learning ability. And the question here is, does learning on the opposite can increase the creation of neuron? And in short, yes. Indeed, some studies have found that some behavior, especially related to learning, may have an impact on the production of neuron, like in the hippocampus. But here again, we have preliminary data, and it's not like something that is fully crystallized, but it seems that indeed neurogenesis can be affected by behavior itself. What do we have again? For now, the last question I have, that will let more question for Pierre-Marie next week, is by P. Lavo about gyrification and creation of pattern of folds in the brain. So the question is, what are the underlying mechanisms, and is that the skulls with its limited size constrain the growing cortex and make it bend? Or is it like a genetic constraint, like a genetic map that plays the folds? Or is it an interaction between the two phenomena? So this is a very good question, because like it's hot debate, and in my lab I have a person, Roberto Toro, specifically dedicated to this question. And it's more near the interaction between multiple phenomena than a clear single mechanism. And if many people have studied the morphogenes, as you saw in the first week of the MOOC, now there's also growing evidence that mechanical constraint on cells can change their expression level. And that's what actually Roberto is studying, Roberto Toro, how the folding of the brain is affecting genes, and on the opposite, like genes could affect how the brain organized, but here it's more like how the spatial and mechanical organization of the brain can change the gene expression. And so you have like a feedback loop here with a lot of nonlinear relationship, as usual. Now you get used to that. But so like a funny thing is that the initial hypothesis would be indeed that we have a growing cortex and a small skull, and that's what make the cortex bend. But you can find a very interesting work by Mahadevan showing that what make the gyrification and the folding pattern is caused by the growing of the cortex, but not necessarily because the skull is constraining it, but more because like tissue grow at different speed. So the outer layer and the inner layer of the cortex are growing at different speed, and that's creating the folding patterns. So there are like many good paper on that. One recently used 3D printed brains in gels and reproducing those kind of gyrification patterns only with gel, without any gene expressed or something, like just mechanically with different growth of matter. So that show that you can have very complex patterns like those of the fold of the brain without a genetic map, and that actually you can have self-organized pattern formation for those kind of phenomena in the brain. And with the work of Roberto on how this can have a causal effect on expression, it's interesting to see that you could have global pattern forming that constrain afterwards gene expression form. So I think I'm done for the question I can answer myself. There is others more related to the course of Pierre-Marie, so I would let those questions for next week. I don't see any person in live right now, but as usual, don't hesitate to ask your question on the forum. And well, I hope you will enjoy the MOOC for the next week, and see you on Monday, same hour next week. Bye-bye.